Okay, so hello everyone, my name is Kai Yu, and I'm from Dr. Kong's lab in UIUC, and today I would like to share the topic about engineering autonomous palm bot with the skeletal muscle. So basically, I will talk about our previous work, collaborate with Dr. Saif and Dr. Zhang Wei in the mechanical engineering, and then there is some future prospect we want to discuss. Okay. So in the very beginning, I want to talk about the tubular heart systems. Basically, in the early stage of our heart development, our heart is not that complex. It's only have two endocardial tubes, and later on they will fusion into together and form one pneumatic ventricle. So this ventricle has a pretty simple vascular network, but they can still pump our blood in a certain direction. And it's same as the open security system in the inside, like cricket. They have an open heart, and they also have a tubular heart. So, but why this quite simple structure can pump this flow into a certain directions is quite interesting. So, scientists start to look into the mechanism into it. So, there's one science paper discuss this mechanism behind this phenomenon. Basically, we can understand that there is an impedance mismatch between these surroundings, so it will allow there's a refraction wave, so we can control the flow into certain direction. Let's use this diagram as examples. If we make a construction at this spot, and it will create a bidirectional wave. And once this wave encounter an area with the different impedance, it will reflect back to the another direction. So that basically they will suck the fluid for a moment a little bit, and once the reflect wave keep go forward, the fluid will go in this direction. So this is the mechanism that this pretty simple structure can create a unidirectional flow. And by inspire of this pretty simple vascular network, our lab collaborated with Dr. Saif to form a pump bot, which is composed of three components. One is the stiff tube, one is the soft tube, and one is the muscle ring. Because we need the tube with different impedance, so we can use the mechanism we mentioned below to create a unidirectional flow. So once the muscle rings on the soft tube construct, it will create like a bidirectional wave, and the wave encountered in the stiff tube will reflect back. So the ring must be asymmetric so that we can create a direction flow in this direction. In this direction. Yeah. So this is the real view as what our device. Basically, we use PDMS to make the skeletons, and we culture the C2, C12 cells in the PDMS mode, so then we can form in a muscle ring, and we combine it with polyacrylamide hydrogel. So then we assemble this hydrogel tube into this skeleton. So that here is our hydrogel tube, and outside is our muscle ring. So later on, when we look into the view on the ring site, we can see that it can contract by itself, which is spontaneously, and also it can contract when we apply some external stimulus like electricity. And if we look into the fluid here, we add some fluorescent microsphere so that we can see indeed this microsphere will moving in these certain directions. So as the mechanism we discussed below, it will go back forward a little bit and then move forward. So when we check in the displacement of this microsphere, we can see that the velocity profile is fit quite well with the parabolic distribution, and also we can see that it also works when we apply some external electricity stimulus. So we are still thinking about what's the future possibility of this pump bot. And one possible way is that we want to combine it with the neurons so that we can form neuromuscular junctions in these systems so that they can contract autonomously and more powerfully. So, but since this tube is suspension in this flow, so it's quite hard to um, make a rigid way to like connect these two, like neurosphere and the tubes. So here is the cross-sectional view, and one possibility I think about is maybe I can create like two panel or pillars in a skeleton in the PDMS skeletons, and later on I can use like soft lithography method to create a reservoir with a certain hold in the bottoms, like they can allow the neuron to go pass through, and then they can innervate with the muscle ring tube. But it's in a pretty early stage, so we're still thinking about how to make this work. And once we can formulate a neuromuscular junction pump, then the promising one promising possibility is that we can make this system into like a drug screening platform, which is like we can make a biomimic circulation system. And we are thinking about that. Maybe we can use this platform for like drug screening for neuromuscular junction disease. As you can see here, if we the medium can pump by the is neuromuscular pumps, and then you will enhance the drug release into the reservoir so that they can interact with the neuron and muscles, and then we can collect this flow back to the loop again. So there's two common neuromuscular junction disease, and one is so-called myasthenia gravis. So this disease is caused by the antibody in the patient. They will block the acetylene acceptor on the muscle so that the muscle cannot receive the signal, so we cannot able to control our muscle. So how to create this scenario or this condition is that we can add in the serum from the patients into like this feedback loop systems, and then we can add the drug in this reservoir and to see which drugs may be a potential can be a potential drug for this disease. Another common disease is so-called amyotrophic lateral sclerosis, which is caused by the injury of neuron or death of neuron, so that the neurons cannot pretty well interact with the muscles. So we can we can get this injured neuron or death neuron from the iPS neuron stem cells from the patient, and then we can co-culture it with the muscle rings. So then we can also think about what's the potential drug for this disease. So in based on this idea, the Saif group already demonstrate like a feedback loop systems, which we created a 2U channel, and there's a nozzle here. So in the video, we can see that this feedback flow can go into a reservoir in a certain direction, and it seems to work pretty well. So later we were trying to like couple this muscle ring with the neurons, and then actually there's a lot of potential where we can think about it, like maybe we can make some like self-tuning biopump, depends on the chemical concentrations in the feedback loop systems. We may enhance the pumping rate or decrease the pumping rate if the muscle ring can recognize some chemicals in this. So this is so I just want to propose some like future ideas about these pumping systems, and if you have any idea or any questions, welcome free to ask me, and welcome free to discuss. And in then I want to thanks Dr. Kong and Dr. Saif to make this project, and also thank you for your listening. I will stop here.
Great. Okay, we have a few minutes for questions, and then also We have 10 minutes for just discussion in general.
That was great. This is Ron. So I have a quick question about the neurosphere and the connection to the muscle strip there. So that you have this in slides, yeah, this slide right here, that you get into an asymmetric situation where there might be more innervation of the muscle ring at the top rather than the bottom. So I wonder whether this asymmetry could be problematic, whether it might make sense to try to create neurons that surround the entire muscle ring, or conversely, can you actually benefit from asymmetry in actuation of the muscle ring?
That's correct. So the best scenario, maybe we can like create a neurosphere like can surround the muscle ring. So, but I still need to think about that way. I do not have like certain purpose to create a asymmetric innovation here. Yeah.
Hi, this is Roger. I had a question on the whether you've thought about linking the pump bot with a vascular system so that you could actually um, generate a vascular flow that could be connected, for example, with another organoid, some other organ type. So you could actually start to generate, physiological type circulation.
Hmm. You mean like we can add some like oligonucleotide in our reservoir so that
I mean, having actually a natural vasculature
uh-huh.
using endothelial cells to grow vessels that would connect the pump bot with another organ structure, whether it be an organoid or some other kind of a construct that would have organ function.
Mm. You mean connect? Sorry, I'm not so sure. Like,
Because that's what we had talked about earlier with Tahir and June.
yeah.
That ultimately what we would like to do is be able to use something like this in like a microfluidic, a microphysiological system, so that
Hmm.
the pump would provide the circulation to other organs. And taking that one step further, if we're thinking about things in the future, the further future, actually developing a natural vasculature that connects your cardiac pump with, say, a liver organ pump or
Yeah, yeah,
some
yeah,
other
yeah.
brain organ, something like that.
Yes.
Doing away with the non-natural parts here, all the parts that you have to basically make out of PDMS right now.
Yeah. So yeah, let's actually the yeah, another good future directions. We're still thinking about it. And also maybe we can make like this for system with a open into an open system so that we can pump this once it can be like they can coexist with the cells, then maybe we can use this pump in like pump some like growth factor or something like making into a biogenerate generate bioreactor so that we can pump some certain chemical or essential chemicals into the certain organ we want. So there's like these potentials we are try keep thinking about it.
Hi, Roger. This is
Sounds good.
June.
Thank you.
So, Roger, this is June. Actually, I actually should not answer your question actually because this is Kai's presentation. But if I briefly actually mention like what we did before, we basically made a kind of a middle-sized endothelial lumen and wrapped that with the muscle rings in order to kind of make a structure you described. We didn't go to the capillary network yet. But at that time, we hope to see kind of a very nice endothelial muscle interactions. But somehow actually muscle actually is too powerful, so therefore it ends up actually collapsing the endothelial lumens. So that's actually was a kind of the result we actually encountered last year. So basically, we are still trying to kind of pursue actually this problem.
Yeah, I guess if you get complete collapse of the endothelium, that could be a problem. But
Yes.
even so, if their circumference doesn't change too much, the endothelium might still be intact. Maybe it's a matter of getting it to adhere better.
Yeah, that's actually a good part. Like we hope to see a kind of a long-term actually effect in order to see what is happening. But anyway, just that actually kind of the work actually was the stopped actually for a moment because the people actually kind of left the group, and then like Kai Yu actually is the very new person, so he actually will also kind of look at this problem again.
We have we have one comment, or it's a question from Iman. Can we use smooth muscles instead?
Yeah, sure. But so some people have shown that we can use like cardio muscles to make into the rings, but we have already compared it, like the construction force produced by skeletal muscles will be stronger than the smooth muscle.
And then an additional, something additional to that question: to prevent the collapse of the lumen beck of skeletal muscle rings. Is that? And Iman, can you can hear us, correct? Are you muted? No. Oh, there we go. Oh yes, it looks like you're unmuted now.
Can you hear me right now?
Yes, you may want to ask your own question just for the because I'm not super familiar with that term.
Sorry about that. No, just because thank you, Kai. It's a very nice presentation. Since the skeletal muscle is very strong, so that it basically collapses the endothelial tube lining beneath. I was thinking like of a system that kind of resembles the original vasculature, where instead of you have a muscle ring that's composed of the skeletal muscles, and that will be strong and collapse the underlying tube, you can just use smooth muscle layer, and it will be more similar to the vasculature, and maybe it will prevent the collapse that you're seeing.
Yeah. Maybe we can try that.
Well, Iman, I can tell you the answer partly. Like the smooth muscle cell is not contracting as powerful as the muscle cell, so we can prevent the collapse, but we cannot get a function of the.
Yeah.
That's another problem.
Ooh, oh, all right. Yeah, that makes sense.
I think the pumping rate will be pretty small. Yeah.
Maybe then pick skeletal muscles are the least active skeletal muscles in the body. So some skeletal muscles are known to be stronger than others. I know that the C2, like the culture skeletal muscles are more available, so it's easier. But maybe then if you can, like, I'm thinking that on top of my head, but maybe like pick a muscle in the body that's known to be the weakest muscle, and then try and culture this one and see if you can solve this problem. This is like an idea. I don't know if it will fit with your project, but just the thought.
Okay. Thank you.
Okay, we've got just a couple more minutes if anyone has a final question for any of our speakers. Otherwise, I just wanted to let everyone know that our next meeting will be scheduled for the week of May 18th for our next virtual research symposium. And let's see, Kaylee, is there, I know it's kind of tough for you to talk right now. Is there anything that you want to add while everyone's still here?
The only thing I would add is the topic for next month is fun things with brain organoids. Is that correct?
Yes, it is.
Yeah, so that will be in the middle of May, and we'll make sure to include everybody on the mailing list.
Okay. All right. Well, I would like to thank everyone for coming, and